the webinar. This is on the science behind the coronavirus vaccine, which is hosted by the Basking Ridge Indian community. My name is Dr. Rajesh Rao, and I would be the moderator for this session. I would like to thank Gautam and Madhvi Pupala and Ms. Prachi Jain. They have been instrumental in setting up this webinar. And this program is organized by the Basking Ridge Indian community, partnering with the local healthcare professionals, experts in their fields. And this webinar is based on our best knowledge that we have and the facts that we know of at present. So there's a lot of data that is evolving. We do not endorse any product or treatment at this point. Please consult your healthcare professional on your personal healthcare needs. And there is a recording of this webinar which will be shared, and permission for this recording has been obtained. A special thanks to our sponsors for tonight's webinar, AT&T Employee Resource Group, the OSS Northeast Group for their sponsorship. Thank you very much. Please join me in welcoming the panelists tonight. Let me introduce the two experts in their fields, Dr. Seema Berry, who has been with us during the peak of the pandemic in April. And we have Ms. Tanvir Kaur, a PhD. Dr. Seema Berry is a board-certified internist and a longtime resident of Basking Ridge. And she has been an integral part of the Hills community for the last 15 years. She is an internist with a focus on geriatric medicine, continuing care retirement communities, and subacute rehabilitation care for the last 20 plus years. Dr. Berry did her MBBS from Lady Hardinge Medical College in New Delhi and her MD residency in internal medicine from Thomas Jefferson University in Philadelphia. She's a director of medicine in two nursing homes, Morris Hills in Morristown and Morris View in Morris Plains. She is also a physician at our nearby Fellowship Village for the last 12 years, and she's an attending at the Atlantic Health Systems Geriatric Center. She sees patients for geriatric consults as well as primary care. As you may recall, she has been in the front line of combating this disease, and she will be giving us an update since our last webinar. Our second panelist is Ms. Vaccine Center. As a note, the listeners can ask questions at the Q&A section of the webinar, and I'll try to answer. We'll try to get that to all the questions, as many as we can. Let me start with a question to Dr. Berry. Can you just give us a quick update on the current situation in our community as you have been seeing in your hospitals? And what is the current treatment that you are giving for the active infections?
Hi, thank you, BRIC, for this platform. Actually, this started by some people asking questions on a casual WhatsApp, and I think this is an important topic. So I'm very grateful for the platform that you guys have provided. And Rajesh, thank you for the kind introduction. So as Rajesh mentioned, we have obviously evolved not only as a community, but you know, as in medicine with this disease, the physicians have learned a lot firsthand, and with a lot of help from our medical community, we are now trying to turn the phase of this disease by obviously first the you know, the know-how, how we are approaching the patients, and of course now the newer treatments and the vaccine. We certainly are not out of the woods. There is no treatment at this moment that is 100% effective, but we are seeing a second peak, and I don't think it's the second peak, it's actually the peak. It is a many more cases in the community, many more cases in the hospital. We are nearly full with our ICUs, and we have I think, peaked at the number of cases that are positive. The difference this time is that we are sort of more prepared. We know how to look for the disease, we know how to look for the complications, and we know how to react early on to somebody who is now spiking a fever. And the other second really big thing is that the testing is much more available. So, we are being able to test people, get positive results much earlier than when their complications start. But the disease really hasn't changed much. We are still seeing a lot of morbidity, a lot of complications, even out 2 to 4 weeks from the initial onset of the disease. So the newer medications that are out there are, they're actually 2 or 3. There is still nothing for prophylaxis. I'm asked this all the time. We think we've been exposed. Can we take something? No, there is not anything out there. There are still sort of articles straggling in about, different kinds of medication. Plaquenil, of course, being on the forefront, but there is absolutely no indication that any of these medications work. The Z-Pak route is also very skeptical. We are not giving that as a routine. There are 3 sort of standard of care treatments now. If patients are have comorbidities, that means they have diabetes, hypertension, but they don't have a sort of disease bad enough that they should be hospitalized. That means they have an oxygen which is above 93, 94%. We are now giving outpatient the plasma, the
antibodies.
The antibodies. This is from Regeneron, and this is actually what Donald Trump got. So that is a push by the hospital because it has just been emergency approved. And what it does is it primes the immune system. By these antibodies, the immune system is already primed and can then react to the virus much more effectively. This is given as an outpatient. It is not given in the hospital. If you are sick enough to be in the hospital, the 2 medications that are being given are the dexamethasone, which is a steroid, which is only given when patients have shown to have a significant immune response and are developing complications from that immune response. So if they're becoming very hypoxic, if their oxygen levels are falling, or if their inflammation markers are very high. The second medication is remdesivir, which is sort of blocks the replication of the virus. It's sort of an antiviral. There have been recent articles from the WHO saying that the remdesivir actually does not make a difference, but there is a lot of backlash to that article in the medical field, because that was an article that was basically from lots of centers, and every center had a very different kind of outcome. So at this moment, remdesivir is certainly being given. But the best thing, again, emphasizing, is don't let yourself get so sick. Make sure you have distancing. Make sure you are wearing a mask.
Okay. Thank you very much for the update. So if you want, we can get started on what you have prepared for the vaccine. What are the different types of vaccine in development, and what are the different, how do you compare these vaccines that are available?
Is this for Tanvir or for me? Okay. So I just want to share.
Just Tanvir can give us the information about how it is approved through our emergency use authorization process.
Mm-hmm.
That'd be good.
Yeah. So Gautam, if you can have slide 1.
I'll do it.
Okay.
Oh, no, sorry, not this one.
Okay, that is Tanvir's, but that's okay.
I got it.
Yeah, that's fine. So the basic vaccines, there are many vaccines. Of course, everybody is talking about the Pfizer and the Moderna vaccine, which is the mRNA vaccine. Most of the vaccines are employing the antibodies against the spike protein. And that's why I wanted to have my first slide, which basically shows you
I'm just looking
the model of the virus. Now, the spike protein is the way the virus attaches itself to the cells. Once that virus attaches itself to the cells, it enters the cells, and that's how it is able to start the infection. So if we can block...
Yeah, can you see that?
No. So if we can block that attaching, that's where we can stop the virus getting into our cells and can immediately stop the illness. Most of the vaccines have sort of homed in on this process, because if we are not allowing the virus to initially enter our mucosal cells in the nasal lining as well as in the respiratory mucosa or the lining of the lungs, then we have stopped the infection completely. So
okay, can you see the slides now? No,
I can't, but
no, we cannot, Rajesh.
Okay,
I'm sorry.
We're seeing your other window.
Yeah,
Different
but
screen.
that's fine. So the two main vaccines are which are now in play are, so we have 85. I think there are 55 vaccines in trials, and I think there are another 85 vaccines in sort of the developmental phase. So this was
Rajesh, yeah, this is the slide.
Yeah, so this is the slide I was talking about. If you look at that, the E protein is the protein outside, the S protein is the one that the way it attaches. Just go to the next slide. I can't move this myself.
No, you can't.
Okay, so I just wanted to show you this. I got my first COVID-19 vaccine today at 5 o'clock, and I am feeling perfectly fine. So I just wanted to let everybody know that. The next slide. So the different vaccines in development are the mRNA Pfizer vaccine, which is in collaboration with the BioNTech. And it has already reached phase 3 trials. FDA has approved it. And I'm going to let Tanvir go through the whole sort of basis of how that was done. The second one, of course, is the Moderna vaccine. And that, again, is an RNA vaccine. And it is also going to, it is also going to get emergency approval tomorrow, actually, is the day when FDA sits down. The third vaccine is the AstraZeneca one, which is a viral vector. And I'm going to explain the two differences, and I'm going to let Tanvir talk about the other vaccines. So the mRNA vaccine is, the mRNA is a small portion of the virus that is codes for different proteins. So this mRNA is coated in a lipid sort of envelope and then introduced into the body. The muscle, when it is injected in the muscles, the cells take up this mRNA. It does not enter our nucleus, so it does not interact with our own genetic material. It remains outside the nucleus and in the cell part. And the body then says, okay, mRNA is there. Let's make the protein that this mRNA piece, it's a small piece of the virus. This small piece can code for the spike protein. So what happens is it starts coding for the spike protein. These little spike protein particles are then excluded out of the cell. As soon as they're excluded out of the cell, they go into the lymph nodes, and the lymph nodes see this virus particle as a foreign protein, and then starts building up this immune response. The immune response mostly is in the T cells, and the T cells are cells that remember. And so what happens is now the body has understood that this spike protein is foreign. It has started a reaction with the T cells. And anytime now this person is exposed to an infection with the real virus, that virus is going to enter and the spike protein is going to enter the cell. The minute the spike protein tries to enter the cell, the T cells and your body remembers it. and starts making all these antibodies that are going to completely fight this virus, and you're not going to get the infection. That to me is an amazing feat, and I think it's just a very new technology. It has never been done before. So I'm going to let Tanvir talk about the different vaccines in development, but really that is the mRNA technology. The second one is the DNA, the viral vector and the DNA. So I'm just going to explain the viral vector, and then Tanvir can talk about the other ones. So the viral vector is a little bit different. That's the AstraZeneca along with the, I think the one in India. So what happens with the viral vector is that the DNA is introduced into the body. The DNA then goes into our nucleus. The DNA then uncoats the RNA, and then the RNA again does the same thing as the Pfizer and Moderna RNA. So it has actually 2 steps instead of 1 step. And that is sort of how the basis is. The other vaccines have, the other viral vectors are basically, sort of particles of a virus that does not infect the humans. So like the rhinovirus that does not affect the humans, the chimpanzee rhinovirus. These viruses are introduced into the body. They are carrying the mRNA of the spike protein of the coronavirus. It's a little complicated. And then these are then introduced into the cell. Again, all these have 2 or 3 steps. The Moderna and the Pfizer have taken away all these extra steps and are able to just introduce the mRNA itself into the body. Again, I want to emphasize, these do not affect our DNA. They do not get into our DNA system. And as far as we know, these are safe, and the mRNA itself gets degraded very fast by the cells, so they don't last. Tanvir, I'm going to let you talk about the rest of the vaccines.
Okay, so Tanvir is going to talk about how this vaccine has been approved for emergency usage, and let me put up your slides as well. Yeah, can you see that now? Yeah.
Tanvir, you're on mute.
Tanvir, you're on mute. Yeah, you're still on mute, or through an email that will give us more details about that.
Actually, Rajesh, I have a very good slide on the side effects, and I just want to bring that up from a clinical point of view. Okay. So if you can go to that. So like Tanvir said, that basically there were 2 published articles. One came out in Nature, and the Moderna one came out in the New England Journal of Medicine. And these phase 3 trials included 43,000 in the Pfizer and 30,000 people in the Moderna.
Okay.
And as she's pointed out, the efficacy, that means 95% people developed antibodies to the virus spike protein. That is what 95% efficacy means. And in the Moderna, it was, I think in the Pfizer, it was 90%, and then they said 95. And then in the Moderna, it was 95%. What Moderna also did differently from Pfizer is that later on, they added, when they did their phase 3 trial, they actually added about a number of patients over 65, because as we know in the elderly, the vaccinations, any vaccine, whether it's the influenza vaccine or any of the vaccines, have a lower efficacy in the older people. So they included older people. And what they saw differently from the flu vaccine to this vaccine was that actually the older people had an increased response. So the T cells, and the reason for that is actually, if you look down, the flu vaccine actually has only a 60 to 75% efficacy. And as you go to the older population, it actually even drops down to 50%. And here we have a 95% and actually a higher response in the older individuals. And the reason for that is because it activated our T cells rather than the B cells. So the B cell is a short acting immunity, which propagates that in the body. And the T cell immunity, which is that is a longer acting immunity. And so the whole question about whether these will be efficacious in the long run can actually be probably answered because the C8, the CD4 and the C8-4 and the CD8 are activated and the T cells are activated. They have a better memory. So 4 months later or 5 months later, if you are exposed to the spike protein again, that memory cell triggers off and the immune response is made. I'm trying to say this in a very
vaccines, and both the vaccines had similar long-term immunity.
So the long-term immunity, of course, as pointed out, has only been followed for, I think the full follow-up was 3 months. And the neutralizing titers were very high and they have not fallen. But again, we still need to keep, those studies going.
And the studies are still going on, is that correct?
Yes, there's 2 years of this. Both the studies are going to run for 2 years. So I think the data would keep on coming every 2 months, 3 months. And then
I think we have a lot of questions from our
go ahead with the questions.
With the listeners. Where can you call to get a vaccine priority based on age and medical condition?
So right now there are even I'm part of the geriatric assessment, and so our office should be actually one of the first few people to get that information, but we haven't. Right now, the hospitals have got the vaccine so that they can vaccinate the healthcare professionals. Okay. And CVS is distributing the vaccine to the long-term nursing home personnel so that the long-term patients as well as the people working there are going to get it. Some of the county healthcare departments have gotten it, but how they're going to, sort of implement it and how they're going to identify who's at risk. There are 3 sort of silos. The first silo is healthcare professionals. The second silo is first responders, so firefighters, teachers, people who work in pharmacies, even people who work in sort of any other healthcare industry are in the second silo, and then patients who are at high risk, older adults, people who've had, comorbidities like cancer, who are immunosuppressed. A lot of rheumatology patients are on immunosuppressive agents. People who've had transplants are immunosuppressive agents, so they are very high risk. So that's like the second silo. And then the third silo is, of course, younger adults so that they don't spread it.
Okay.
Yeah.
So there's a question for Tanvir is, which of the COVID vaccine uses a newer DNA technology?
There is no DNA technology.
No, it's all mRNA.
It's mRNA.
No, there is an activated in non-replicative DNA. That's the AstraZeneca is using the non-replicative DNA. There are actually 2 or 3 vaccines that are using the non-replicative DNA vaccine, and I'll tell you which ones they are. So AstraZeneca has 2 out, the Ensemble phase 3 trial, and then the Novavax is using the adjuvant vaccine. So the AstraZeneca 2 trials are using the DNA subunit. Yeah.
So if both are available, the Pfizer and Moderna, which one should one get?
No, so the Pfizer and Moderna both are RNA. They're not the DNA vaccine.
You can get only one. You can get only one of them.
Sorry?
You should get only one of them, not both of them at the same time.
No, they are the same technology, and you will not be allowed. You will get either one or the other.
Correct. So there is no point in getting both of them. So
right.
Is it true that the success of vaccine is only 95%? So 1 in every 20, 95%. So 1 in 20 may get vaccine injury, unlike current vaccine. 1 is okay. So the question is it true the success of a vaccine is only 95%?
The 95% is actually very high. Just to put into perspective, the flu vaccine has only a 60 to 75% efficacy. Okay. So 95% is actually probably one of the highest we've seen in any vaccines.
Yeah. So again, the question I think is a little skewed, because the efficacy of the vaccine is 95%, means that is not that 5% have adverse reaction. So I think that's the question, was whether the 5% have an adverse reaction. So the answer to that question is that the 5% are not adverse reactions, but 5% is they are not effective, like they are not going to get immunized totally. That's the
so the 95% efficacy means that 95% of the people who got the vaccine
Correct.
have developed enough antibodies to not get the disease.
Correct.
And a 5% are people who may
Correct.
get the disease in spite of getting the vaccine.
Yeah, so that was a clarification that I made,
Right.
was that the 5% is not the people who have adverse reactions. So adverse reactions are very minimal.
Yeah, so if you want to put my adverse reaction slide, you can. So the adverse reactions are, what they've seen in both the trials is that the adverse reactions were non-threatening. They did not have any life-threatening reactions. The AstraZeneca one had the transverse myelitis, which is basically inflammation of the spinal cord. That was not seen by the mRNA. The local reactions that were noticed by the mRNA, Pfizer, as well as Moderna, were injection site pain, fever, pain in the muscles, and generalized nausea, maybe fatigue. But most of these lasted for maybe about a day or two, and none of them were long-lasting effects. None of them were life-threatening effects at all.
Okay, so the next question is, once you take the vaccine, how long will it be effective? And so I think that is another question, is between the two, you can give the information also, is between the two vaccines, the gap between the first and the second one. What is the gap in between the first and the second?
So in the Moderna, it is 21 days, and that's my slide is there. And in the
28, isn't it?
28 days, and in the Pfizer, it's 21 days.
21 days. So it's more, so 3 weeks for Pfizer and 4 weeks for.
Yeah, and the reason for that is not, so the reason for that is that the first injection basically primes the body to start recognizing. And then when you give the second dose, which is a higher dose, that dose then is so effective because the body has been primed to produce and understand that this protein is foreign and start producing these antibodies. So that second dose really is the one that is the most effective. So please understand, whoever takes the vaccine, you've got to get the second dose, because the first dose is not enough.
Okay. So do we have to take the vaccine every year, Tanvir?
We don't know yet.
Okay.
We don't know yet.
Well, the next question is, since we have choices of different manufacturers, which one should you take and why?
So as Tanvir pointed out, the Moderna was in conjunction with the National Institute of Allergies and basically, National Institute of Health. The Pfizer vaccine was with a company called the BioNTech. The Pfizer vaccine had obviously the first results. They had 43,000 people they tested in. The Moderna had 30,000. To my mind, today I got the Pfizer vaccine. I really didn't see any difference. If I was offered the Moderna, I would have taken the Moderna. I think the more important part is that we get the vaccine. What is available, we get it, whether it's the Pfizer or the Moderna. The others, of course, are not available yet. So I don't think I would have any preference. Again, 92% to 95% efficacy, similar side effects. Both the trials had actually very similar side effects. The problem is storage. So if you are confident that the place you're getting it has good storage in terms of refrigeration storage, then go ahead and take any vaccine. If you're not confident of the area you're getting it in, and you don't think that the storage that they are doing is going to comply with what is necessary to have the vaccine sort of in an activated form, then maybe try to take the Moderna if you can. But hospitals are very well equipped with keeping the storage the way they have to in refrigeration.
Okay, so the next question is, when do you think the seniors in our community will get the vaccination?
So the seniors in our, the seniors in the rehab are getting it next week, starting the 21st.
The one who are hospitalized, the one who are they are in the rehab center.
Yeah, they are already in rehab centers. And as I said, I am part of the geriatric assessment center, and our office has not heard yet. So we would be the first people you are hearing, because we have only geriatric patients as outpatient population, and we haven't heard yet. But I assume that it's going to be like first or second week of January.
Okay.
You answer it, but I'm just going to add a vaccine that had started being developed and used in the 1990s. It was meant to be used for a cancer vaccine, and actually has also been used for an influenza, trial influenza. It has been tried. Tanvir, you can answer that too.
So it's been, again, it's...
RNA has been trialed in different trials for cancer vaccines, for Ebola, and which was not successful, and then for a previous influenza that has been trialed in humans before.
Okay, so there's a question: how do you manage the risk of what we do not know? What we do not know means basically ignorance of what we don't know, given the large disparity in the trial participants versus the size of, the broad population.
There was no disparity. I don't understand that question. The trial group in both groups, 43,000 people in Pfizer and 30,000 people in Moderna, reflected the community. They started from age 16 to actually age 95, and everybody in between. And the big chunk of the 43,000 people were people aged between 35 to 75. So as I said, the Moderna vaccine also actually made sure that they had people who were immunocompromised, who had hepatitis, and who had transplants. So this reflects the community. There is no your question about, it's not reflective of the community is not true at all. It is very reflective of the community.
Were there any neurological complications like GPS, Guillain-Barré syndrome?
There was actually, surprisingly, most vaccination trials have some neurological. I know the AstraZeneca has had transverse myelitis, and the trial was stopped, I think, sometime in September. There was one case. I'm not sure if they had more cases. But in the Pfizer and the mRNA technology, they did not see any neurological cases, which is surprising.
So how does it cover for the mutations of the virus with one vaccine?
So it doesn't matter whether the virus mutates. So the virus will mutate in its other part of its DNA, but the way it attaches to the cells is from the spike protein. So whether it mutates or not, it doesn't matter. It will never mutate, because that is one of the most important proteins it has. So as I showed you the slide first, there's a spike protein, and then there's the envelope protein. Right. So these are very inherent parts of the virus particle, which it cannot mutate, because then it'll become a totally different virus. So if it mutates in terms of how much it causes an infection, or how infectious does it become, or it doesn't matter. The spike protein is always going to remain. And mostly all these vaccines, whether it's Moderna, Pfizer, AstraZeneca, viral vector, or whatever it is, are using the antibodies produced against the spike protein. So it can mutate, it doesn't matter, but the spike protein will always remain. And that's really what antibodies are against.
So if you already have antibodies, should you still get vaccinated? Should you get vaccinated if you are already positive and antibody is positive?
Right. So they're not taking into account people who have had the vaccine, have had the viral infection or not, or have antibodies. They're not checking for that. And the reason for that is that if you develop a antibody response when you have infection, you can lose that antibody response as the times pass. But when you have a vaccination and you have an antibody response to that, it is usually augmented. And the reason for that is that when you get the vaccine, the response of your antibodies, the number of antibodies you make against that one spike protein are really outnumber what you would do in just an infection. So they also use adjuvants, which they haven't used in this, but usually the response to a vaccine is much more than the actual infection itself. Tanvir, if you have anything more to add to that.
No, you've covered it perfectly well.
So is the antibody that you get from the virus is towards the spike protein when you get infected?
Yes.
Yes, it is towards the spike protein.
So it is the same antibody to the spike protein. Okay.
Yeah.
There's a question. Do you think the people who have already had, okay, it's the same question. So even if you're infected, you should still get the vaccine. Let me get to the last few questions. Lifetime, nobody knows that. What are the side effects we covered? How does we know the facility? Okay. So how do you know that that vaccine that you got, SEMA, has been stored at minus 70 degrees?
Because I trust my health system.
So I can add to that. I can add to that. So
that is the answer to one of the questions.
Yeah, I can add to that. So it's a very, very stringent.
Do you think the general general population will get the vaccination? What is the timeline for that one?
So I think right now, it really depends on how much we are going to be able to, how much these companies are going to be able to, how fast they're going to be able to manufacture, how many vaccines are going to be sent out of America. But I think the projections are that till January, we'll vaccinate our healthcare workers. From the beginning of January to the middle of January is going to be the nursing homes and the long-term patients, and then the emergency workers, the teachers, and then I think by March we're going to be able to get it through CVS.
Okay, so the question is whether there is vaccination needed for people who are immunocompromised.
Absolutely, very much so.
You have to give it, right? And what?
Yes.
Yes, it has to be a separate arm.
Yeah. So there are some questions which have already been answered, but I'm going to go through that. Are Regeneron and Remdesivir treatments now available to everyone who's infected?
So again, there are. I'm just going to sort of answer that question. There are criteria for both Regeneron as well as Remdesivir. The Remdesivir criteria are quite strict. You have to be hospitalized. You have to have an oxygen level below a certain amount. You have to have normal kidney function, and then you will get the Remdesivir. For the Regeneron, you do not have to be hospitalized. You have to be referred by a physician. You have to, usually most effective in the first 10 days of the infection, usually not very effective after 10 days. And you should have some comorbidities like immunosuppression, diabetes, hypertension, or age. And you should be having a oxygen sort of above 94, 95%, so that you're not then hospitalized for Remdesivir.
So there's another question regarding ivermectin as a potential treatment, recently used by ICU doctors.
Yeah, so the same physicians who basically pushed for dexamethasone as treatment have been looking into this. This is being used in Asia right now. There are no studies, even anecdotal studies, which are coming out. There may be a lot of interest in it in the next few months. Mm-hmm. It is an anti-malarial drug, and it is being used in Asia, and I think it is being used in Germany quite a lot against the virus, and they've seen some really good sort of outcomes. In terms of decreasing the amount of days infected and the severity of the disease.
So again, the question is going to be a little bit challenging in the next few months is when you get 3 different vaccines in the market, which one would you choose? Is there any data on that? Or I don't think there'll be any information for us to make a choice on.
Yeah, I don't think so, but I'm looking at the technology and I'll tell you one thing. The mRNA technology is here to stay. It
Okay.
is going to revolutionize the way vaccines
So
are
the
done.
mRNA you're talking about is the Pfizer and the Moderna.
Yeah, the Pfizer and the Moderna. It bypasses so many steps which the viral vector and the non-replicative DNA and the nanoparticles are using. So I think mRNA technology is here to stay. And I just wanted to just bring one point up. So the new Shingrix vaccine, which is against the zoster, is so effective compared to the old vaccine. The old vaccine was the live attenuated. The new zoster vaccine is actually a particle, again, the glycoprotein coat, just the same way like the spike protein, there's a protein coat, and the antibodies are mounted against that. And that technology has been so sort of efficacious. It is so amazing, and how long the efficacy lasts is also very much more superior than the previous vaccine. And I just think these technologies are really here to stay and completely will revolutionize vaccinations.
Okay, there are a few more, I think, in a different chat group. Vaccines can cause, how do you prevent an overreaction, like of an immune response, like a cytokine response with a vaccine?
I'm going to answer it, and if Tanvir, you have something to add, you can do that. So they really, in the 95% of people they saw, they did not see a immune, because it is not the whole virus. Okay. You are only producing an antibody to the spike protein. So you're really not producing it to the whole virus. And so your immune system does not go into that hyperdrive that they see with the viral infection, because it's only the spike protein that is creating the antibodies, and the T cell antibody response is much more longer acting, as well as a much more stable response. It's the B cell response, which is the shorter acting and much more hyperactive response that you see in these infections. So the antibody response here is more related to the T cell, which are two different types of antibodies in the blood.
And any between flu shots and can be taken together or separately? Flu shot.
Yeah. So right. So vaccinations can be taken together, and I think I'm going to let Tanvir answer that too. But I think that they definitely can. They do create a different response. As I just said, the antibody responses are different. The immune cells that are activated in the blood are different. So will they be given in the same syringe? No, they will be given separately. Like when you are young, you get 3 different shots, you get in different syringes. So because the mucosal sort of receptors where the injection is given, that particular immune response right away is different in different vaccines. So if you have anything more.
Just a quick question for Seema as well. Which organs are affected besides the lungs by the COVID virus?
The COVID virus right now has affected nearly every organ. We have lungs, of course, are the first target, but we have a lot of neurological changes. We have vascular changes. We have heart is affected, and globally, of course, sepsis affects every other organ. So everything.
Excellent, excellent. So I think I'm just go through this. There were a few more questions in the last. I'm not sure I can see them. I don't know how. Let me just check, make sure that I got. I think we covered most of it. I think we have covered. So there are what percentage of people are infected and have been hospitalized. I think that has been covered also. Okay.
Yeah.
Good. Thank you.
So I just want to add one thing. I want to encourage everybody to go out there, put the fears aside, and go and get your vaccine, because we need about at least 75% of the population to be vaccinated before we can eliminate this disease. And I also just want to say, just shout out to people like Tanvir, who are working behind the scenes, who are so dedicated and have done such an amazing job that in 8 months, we have not only been able to identify a very, very safe vaccine, but something that we have been able to manufacture, produce, and then bring it to the public, like me. You know, when I was getting my vaccine today, I can't tell you how proud I felt to be part of this amazing system. And it is just unbelievable what humanity has been able to do. And as Tanvir pointed out, a combination of all the countries together, and people from the medical field, people from the research fields, people who have, you know, just dedicated their time and effort to this. And I just have to say, it's just an amazing feat.
Yeah. Tanvir, do you have any closing statements? You're muted again. Thank you very much to the panelists, both of you. You did a great job on giving us a lot of information. I know this has been kind of an evolving process, and I think getting a clarity on what's going on at this point and what we are looking forward in the next few months will help us in making the decision what we want to do. And I'm sure we're going to do, I think Gautam is definitely going to pitch in, and we might be able to do one more on once we get a little bit clarity on what's going on, especially for distribution of the vaccine to everyone else in the community, because currently as the different tiers that have been formulated by the government is to have tier 1 is the medical community and the first responders. Second is the policemen, the first responders from the community, like firemen and teachers. They are going to be the second one after the nursing home residents. And after that would be the age dependent and medical condition dependent general population, and eventually everyone else. So I think that is what we have so far. And I think once we get a clarity on,
so I guess, you know, maybe good to know.
Good to know. So that's a good thing. Again, thank you very much, both of you.
Thank you. Take care, Rajesh, and thank you, BRIC.
Great.
Thank you. Yeah, thank you, BRIC.
Everybody else, please stay safe and enjoy the snow. Don't shovel it too much, okay?
And yeah. Thank you, BRIC, for this platform.
Thanks. Thank you.
Thank you, Gautam. Thank you.
Really?